Okay, we've covered C3 photosynthesis and all its different parts. Now let's look at C4 and CAM photosynthesis. And in C3 and C4 photosynthesis, the plants have evolved to try to deal with the problem of photorespiration. And the key is that the plants want to do photosynthesis, use these chloroplasts, right, in the proper conditions. So it wants to do this photosynthesis, this chloroplast. It wants to have low oxygen and high CO2, and the concept is that in that way you reduce the amount of photorespiration. Now you see here that you have the mesophyll cells that we looked at in the PowerPoint. You have these bundle sheath cells, and then you have the vascular bundle. This is where the fluids go up and down the plant from its roots to its leaves. Now when you're looking at C4 photosynthesis, the only real difference is the use of a 4-carbon molecule instead of a 3-carbon molecule that you see used in C3 photosynthesis. And the idea is that by first capturing the CO2 using this enzyme called PEP, P-E-P, the plant is able to take that carbon dioxide and change it to malate, and by changing it to malate, a 4-carbon malic acid, a 4-carbon acid, it's able to take it and bring it into a deeper tissue where there's less oxygen and more CO2, therefore reducing the amount of photorespiration. Remember, photorespiration is detrimental to the plant because it cannot use oxygen to make sugar, and on top of that some toxic chemicals are made. So here in the inner cells, we're able to the plants are able to make the sugar that they normally make and transport it out and make whatever biomolecules they need to make. So G3P is made ultimately here as well, but first you have an intermediate called malic acid in order to put the carbon dioxide into an area where there's less oxygen. So the malic acid actually turns into carbon dioxide again here in the inner tissues. Now CAM photosynthesis is done by plants and other succulents, specifically cacti and other desert plants, because in CAM the conditions are such that there's just too hot for the plant to open their stomata, not stroma, the stomata in the desert. So what they do is at night they open up their stomata and allow CO2 in and oxygen out. Again, PEP is used to take CO2 and make it into malic acid, and it just puts the malic acid in a vacuole. Now, a vacuole, remember, is an organ in which the plant can store various materials. And then during the day, the stomata are closed, and the malic acid then is released and becomes CO2, and the CO2 can enter the Calvin cycle by being taken up by rubisco. And again, in this case, there's more oxygen and more carbon dioxide, less oxygen. But what's key in CAM photosynthesis is that the plants are not losing as much water as they would if they opened their stomata during the day. So CAM photosynthesis it separates photosynthesis into 2 parts during day and night, and it adds an extra step of turning into malic acid, a 4-carbon sugar. And where C4 photosynthesis really focuses on separating it by place, where we're dealing with it's going to do it in the bundle sheath cells and captures the CO2 in the mesophyll cells. And C3 photosynthesis just does it as it comes in. C3 photosynthesis is the most used on the planet, but in places where there's tropical regions, where there's a lot of sunlight, a lot of water, you're going to do a lot of C4 photosynthesis. It's more efficient and less photorespiration is done. And photosynthesis is done in places where there's desert, again separated by time, not by place, in order to reserve their water supply. All right, I hope this helped. Have a good one.